Hello everyone, welcome to NEET YUG. I am Arun Kumar. Here I am going to explain non-cyclic photophosphorylation, or it is also called as Z scheme of electron transport system during photosynthesis. What is photophosphorylation? Photo means light, phosphorylation means it is the process of synthesis of ATP from ADP and inorganic phosphate, phosphorylation. Got it? So with the help of light. Why it is called as non-cyclic? Here the electrons are required for the synthesis of ATP and NADPH. Once the electrons move from the systems, like you will see around two systems, the PS2 and PS1. Normally electrons move from PS2 to PS1. Once the electrons are received by the PS1, it's not going to transfer the same electrons back to the PS2. So there is no coming back of the electrons that to PS2 system. Got it? So it is lost. So the movement of electrons is always in one direction. So it is non-cyclic. At the same time, why it is called as Z scheme? It looks like Z. Z scheme means the movement of the electrons. If you see the path of the electrons, it will be like this. Okay, normally from low redox potential, that is, I'm talking about electrons, to the higher redox potential. So it will be in downhill manner. Got it? Making it Z in appearance. That is Z scheme. Where exactly this photosynthesis takes place? Normally photosynthesis takes place within the mesophyll cells of the leaf. Let us take this as the mesophyll cell. What you see is the green organelle, that is the chloroplast, where exactly the photosynthesis takes place. Why? Because it has got certain systems present within it, which normally takes the light and convert it into ATP and NADPH. These are the assimilatory power. Got it? If these systems use sunlight to produce ATP and NADPH, this is called as light reaction. Okay, and again what's going to happen? The ATP and NADPH which is synthesized during a light reaction is going to be like used in the synthesis of sugar within this chloroplast. That is dark reaction where sunlight is not required. Got it? So you see two phases: light reaction, dark reaction. Here I am focusing mainly on a type of light reaction, that is non-cyclic photophosphorylation. How exactly ATP and NADPH are synthesized within the chloroplast. So this is the chloroplast. Okay. Now I have got one more model, that is, you could see what is there inside the chloroplast. Outer membrane, inner membrane of the chloroplast, and this is the stroma of the chloroplast, the liquid portion, and you could see some coin shaped structure which are piled up to make the grana. This is called as the thylakoids. Many thylakoids are there. Each thylakoids or each grana, so this is also grana, this is grana. Many granum now are connected by a long slender like structure called as stroma lamella. Here within the thylakoid, non-cyclic photophosphorylation occurs. Within the stroma, you see this, right? The bridge. Within the stroma lamella, you see the cyclic photophosphorylation where ATP only synthesized. But in non-cyclic photophosphorylation, ATP as well as NADPH will be synthesized. This is the two major difference between cyclic and non-cyclic photophosphorylation. Okay. Now, let us just imagine now the entire board, the green board, as the stroma of the chloroplast. Stroma of the chloroplast, where exactly the non-cyclic photophosphorylation occurs. That occurs within the thylakoid, which occurs within the coin-shaped structure, right? That is thylakoid. Okay. In the thylakoid, where? In the thylakoid, it is in the membrane of the thylakoid. Okay. So I'm going to draw the membrane. This is the outer membrane which is now facing the stroma portion of the chloroplast, and I'm going to draw the inner membrane now which is facing the lumen, that is the center of the thylakoid. Okay, I could have drawn in a circular manner, not required. Okay, so this here is the lumen of the thylakoid. Lumen of the thylakoids. Okay, got it? Now for the photosystem, that is actually for the phosphorylation to occur, what is required is we need systems. We need two systems. The one system which is present here, this system is called as, let me write here, the photosystem 2. If you see physically, the photosystem 2 is some portion here, you could see some portion is facing the lumen. That means it is already embedded towards the lumen of the thylakoid. Got it? You could see that. Okay, towards the lumen of the thylakoid. Another system is also required. So I am using the pink color. This is situated slightly away from the inner membrane of the thylakoid, see, but it is facing some portion of this system, that is PS1, the photosystem 1 is facing the stroma portion. Okay, so now in the stroma some components will be there, that is iron sulfur protein. It is also called as FES protein, which is present in the stroma, which is very important for PS1 to synthesize NADPH. Okay, and then what is required is we need ferredoxin. We need a ferredoxin. I write FD. Then we need one enzyme that is called as FNR. What is this FNR? Ferredoxin. Okay, so same ferredoxin, NADP reductase enzyme. Reductase enzyme. FNR. Ferredoxin NADP reductase enzyme is present only in the outer, very close to the outer membrane of the thylakoid. It will not be seen in the stroma lamella. That is what in non-cyclic or in cyclic photophosphorylation, you will not see the synthesis of NADPH. Only ATP is synthesized, because for the synthesis of NADPH, this enzyme is must. Got it? Okay, and the FES protein is also must. Ferredoxin is also must. And in the stroma, you see NADP, okay, that will be floating in the stroma. 4 components are required. Okay, what is there in the lumen? Okay, inside the lumen we have got water. Let me just write 2H2O. Okay, so when the sunlight falls on the thylakoid membrane, and obviously it will also reach the lumen, water molecules also split, that is 4 protons, 4 electrons and 1 oxygen molecules will be liberated. Okay, this mechanism is called as splitting of water. Splitting of water. We just write it, splitting of water. The other name of this is oxidation of water. Oxidation of water. One more name is, let me write here, the other name is oxygen evolving complex, because 1 oxygen molecule is produced when the water splits. Oxygen evolving complex, OEC. It can also be called as water evolving complex. Got it? So it has got different names. Just remember it, OEC. Now, what is required now for the activation of PS2 and PS1? The sunlight. Sunlight. What kind of sunlight? Red light is required. Let me just write, this is now the sun and the light. The light is, for the activation of PS2, we need 680 nanometer of red light. So I'll write here, red here. And for the activation of PS1, we need 700 nanometer. That is also red light. Okay, fine. And now the sunlight also falls on this lumen, and as a result, the water now splits to we get protons, electrons and 1 oxygen molecules. Okay, now for the splitting up of water, like to synthesize 1 oxygen molecule, how many oxygen molecules are here? Only 1. Okay, to get only 1 oxygen molecules, we need 8 photons. So how many photons are required? 8 photons are required. And this is called as quantum requirement. Okay, let me write here the quantum requirement. Quantum requirement means to synthesize 1 oxygen molecule, how many photons are required? 8 photons are required. This is called as quantum requirement. Okay, let me just write what is quantum yield. Quantum yield. Quantum yield in the sense, to get 1 oxygen molecules, we need 8 photons. That is nothing but, if we convert it into percentage, that is 1 by 8, you get 12% of the photons required to get 1 oxygen molecule. This is quantum yield. Just remember it. Okay. Here, one more thing is, how many normally, like you see 1 oxygen molecule synthesized? Okay, so that is actually very important, because when the water splits, it's going to get split into 4 protons and 4 electrons. Correct. These electrons are very important for the synthesis of ATP and NADPH. Very important. Got it? Okay. Here, let me just write what is red drop. Let me just write it. Red drop could be asked in your competitive examination. It is also called as Emerson'seffect. Emerson's effect. Okay, Emerson's effect. red drop or Emerson's effect. Here Emerson observed that if the wavelength of light, that is red light, if it goes beyond 700, if it is excess more than 700 nanometer, the rate of photosynthesis will be reduced. Okay, so rate of photosynthesis will be reduced if it exceed more than 700 nanometer. He observed that in PS2, that is 680 nanometer, the rate of photosynthesis will be more compared to in 700 nanometer of the red light, that is in the PS1. Got it? So more the nanometer, that is the 700, if it is beyond 700, then obviously the photosynthesis rate will be reduced. He observed it. That is called as red drop or Emerson's effect. Note this point. Okay. Now between PS2 and PS1, what is there? You see a system that is called as electron transport system. Let me just use white color here. This is now electron transport system that is present between these two photosystems. Okay, within the electron transport system, what you see is, okay, so let me write here ETS, electron transport system. The first within this ETS, what you see is the electron acceptors and the carriers. Okay, the electrons move from this system to this system, so we need some components. They are actually electron carriers. The first electron carrier, which is called as pheophytin, let me write here pheophytin. Note one point here, the pheophytin is non-mobile electron carrier, non-mobile, it will not move. Okay, second one, after pheophytin you see plastoquinone. Plastoquinone, this is the mobile electron carrier. Note this point, a mobile electron carrier. Okay, next is cytochrome B6F, which is required, which is also electron carrier and even acceptor too. Next one is, the last one is the plastocyanin. The plastocyanin is the, again, which is the mobile electron carrier. This is non-mobile electron carrier. The plastocyanin will be situated very close to the PS1. Got it? Okay. Now let us imagine sunlight falls on the thylakoid membrane and the PS2 now receives. What is there in the PS2 or PS1? Both are same. Chlorophyll A will be there, that is the major photosynthetic pigment. Along with that you see chlorophyll B and xanthophylls and carotenoids. These are accessory photosynthetic pigments. Okay. The chlorophyll A, when the water splits, when the 8 photons hits this water, it will split into protons, electrons and one oxygen molecule. Okay, that is quantum yield. These electrons are now present in the lumen, which will be accepted by the chlorophyll A. Got it? Chlorophyll A, which gets excited. So chlorophyll A now gets excited. Got it? Now what is there inside the chlorophyll? Electrons are there. Okay, it cannot hold the electrons for a longer time. It has to transfer these electrons to the ETS, which is the first system in the ETS, that is pheophytin. Let me just write pheophytin, a non-mobile electron acceptor or carrier. Then it will be transferred to the plastoquinone. Then it will be transferred to the cytochrome B6F. Then it will be transferred to the plastocyanin. That is also mobile electron transport. PC and PQ, which are mobile electron carriers. Got it? Whereas pheophytin, non-mobile. Okay. So when the electrons are moving, here the electrons normally move from its lower redox potential to the higher redox potential. Just remember this, which makes or which looks like, the electrons are moving in downhill manner, because the electrons move from its lower redox potential to the higher redox potential. That is from pheophytin to the PC, from its low to high. Got it? So this is Z scheme. It is like downhill movement. Okay, which is making in Z in appearance. Okay. Once the PC now receives the electrons, it is accepted by the chlorophyll A of the PS1. Now it transfers the electrons, it gets excited. Okay, then these electrons are accepted by the FeS protein, that is iron sulfur protein. Okay, then it will be accepted by the ferredoxin. Then comes the enzyme, that is FNR, ferredoxin NADP reductase. And here what's going to happen, the NADP, how many are there? 2 NADP are there. It's going to be reduced to form 2 NADPH plus H. Okay, so that means we have got the energy. So this is actually the flow of electrons to synthesize 2 NADPH. Got it? Okay. So in this electron transport system, when the electrons are moving within the electron transport system, what's going to happen? ADP and inorganic phosphate gets converted into ATP. This is phosphorylation. The synthesis of ATP from ADP and IP, that is phosphorylation. See, the movement of the electrons will be in only one direction, correct? So these here the electrons are accepted by the FAS protein, done. So electrons are not going to come back to the PS2, lost. Again we need the water, new water to split and release the electrons, and these electrons will be picked up from PS2 like that. Got it? Okay. Here I am going to explain how exactly ATP is synthesized within the electron transport system in non-cyclic photophosphorylation. Even it can be applicable to the cyclic photophosphorylation too in another concept that is called as chemiosmosis. Chemiosmosis, how exactly ATP is synthesized within the membrane of the thylakoid. Okay, so we understood this is all about non-cyclic photophosphorylation. Remember, very important, even it was observed by Hill, we can also write this reaction as Hill's reaction. Hill's reaction, note this point. See how many names it has got. Red drop, Emerson'seffect, quantum yield, Hill's reaction, non-cyclic photophosphorylation, Z scheme of photophosphorylation or electron transport system. So that's all about from this session. If you understood the concept explained by me, like and share and subscribe to our NEET YUG channel. Thank you.